We were discussing about transplantation, and in the transplantation, because of the mismatch of the MHC complex, the rejection of the graft takes place, and therefore precautions have to be taken to prevent the graft rejection. Now we'll talk about today the immune tolerance, how the tolerance develops, and what is the role of immune tolerance. We know the conventional immune response, how it happens. When an immune cell, that is the first diagram, recognizes a foreign antigen, it gets activated, that is whether it is T cell or B cell, and that undergoes clonal proliferation to develop the immune response against that particular foreign antigen. Now, the tolerance is generally developed against the self antigens. There are two types of tolerance, that is central tolerance and peripheral tolerance. The central tolerance will develop in the primary lymphoid organ when the selection of cells is taking place in the primary lymphoid organ, that is thymus and bone marrow. As far as T cells are concerned, it is positive selection. As far as B cells are concerned, it is negative selection. So whenever the T cells and B cells are undergoing maturation in the central lymphoid organ, that is B cell maturation takes place in the bone marrow, whereas T cell maturation takes place in the thymus. In this process, during the maturation, the cells have to come in contact with the self antigen first. If they don't come in contact with the self antigen, cannot be selected. The second contact should have to happen with the non-self antigen, but the first contact always with the self antigen. When the self antigen contact takes place, those cells are selected in the case of T lymphocytes, and those cells which are selected are called as positive selection. Whereas in the case of the B lymphocyte, it is called as negative selection. And in the negative selection, what happens with the B lymphocyte? Those cells which show strong affinity to the self antigens, are eliminated by apoptosis. That is called as deletion. So they are eliminated by apoptosis. Similarly, in the case of T cells, in the negative selection, if the cells come in contact for the second time with the self antigen, they are eliminated in the thymus. So that process is called as deletion. And the thymus and bone marrow process of development of tolerance, making the immune cells to understand and learn the self antigens so that they don't develop immunity against self antigen, is called as central tolerance. Now, what is peripheral tolerance? Peripheral tolerance takes place in the peripheral lymphoid organ. The peripheral lymphoid organs are secondary lymphoid organs. That is lymph node and spleen, or probably tonsils. Okay, so these are the secondary lymphoid organs. The cells that leave the parent tissue, that is bone marrow, that is by B cells, and thymus, that is by the T cells. After they leave their origin and they come into peripheral circulation in the blood, they will enter the secondary lymphoid organ. And in the secondary lymphoid organ, they have a chance of contacting self or non-self antigen. But in the after the selection process, that is in the central lymphoid organ, if they come in contact once again with the self antigen in the secondary lymphoid organ, they will undergo anergy or elimination once again. Anergy is nothing but non-activation. They cannot get activated against the self antigen. In the lymph node or in spleen or tonsil, if they come across once again with the self antigen, they will undergo anergy and get eliminated. There is also another regulatory mechanism in the secondary lymphoid organ. There are T regulatory cells. The T regulatory cells secrete immunosuppressive cytokines. And the immunosuppressive cytokines like IL-17, IL-10 is in large quantities secreted by the T regulatory cells, will prevent the T cells from getting activated against the self antigens when they come in contact with the self antigen. That happens in the peripheral lymphoid organ. So that is called as suppression. So the T cells are suppressed from getting activated against the self antigen. So this is how the tolerance occurs. Now, how can we demonstrate the tolerance by experimental means? We'll see little later. Before that, we'll take the example of development of tolerance naturally in some cases, and first example that comes is from free Martin calves. They are the young calves which are developing in the uterus of the cow, and they are called as free Martin calves because they were identified by the person called as free Martin. Okay, and this is the natural immunological tolerance. Here, just like the way in human beings, the identical twins develop in the same uterus or fraternal twins develop in the same uterus. Okay, whether it is identical twins or whether it is fraternal twins. Fraternal means dizygotic twins. When they are developing in the uterus, those developing embryos will have separate placenta. They will have separate placenta. They will have separate umbilical cord. Only in a very rare instance, they will have single placenta, okay, single placenta and bifurcated umbilical cord. Okay, so if that happens in the case of human beings, okay, that is the single placenta, and but here what is shown in this diagram is the single placenta, but the two umbilical cords are there. Doesn't matter, it is also okay. But instead of two umbilical cords from the placenta, if one umbilical cord emerges and it bifurcates into two and goes to two fetuses, that is also possible. It happens in the cattle, it happens in human beings, and in the cattle they are called as free Martin calves. Now here in this example in the picture, they are shown two calves connected to the same placenta, single placenta, through independent umbilical cord. But remember, the blood vessels, that is arteries and the veins that emerge from the fetus, which go into the placenta, all right, to receive the nutrition from the placenta, are diffused in the placenta. They are distributed in the placenta. Okay, so therefore, the antigens that are of origin in this calf will have a chance to diffuse in the placenta here. Same way, antigens of this calf also have a chance of diffusing into this placenta. So therefore, when those antigens diffuse, there is equal chance that those antigens can once again be sampled by both of them, irrespective of their origin. All right, I hope you are understanding that because the umbilical cord is connected to the single placenta, whatever the antigens of one of this calf which are coming in contact in the placenta can also be sampled by the other calf. So imagine that situation becomes much easier if the single umbilical cord was originating from the placenta and bifurcating into two and joining these two fetuses. Then what would have happened? The artery and the vein would have come up to a particular point as individual single and then bifurcated. At that time, what would have happened? The antigens of both of these would have got mixed in that artery and the vein in the placenta because of the single umbilical cord. Whatever may be the case, now when these calves are born, okay, even though if they are not identical twins, they are fraternal twins, they are developing with the attachment to single placenta. Therefore what happens is, now let us take this example here, this free Martin calf. Two are of different colors, they are not identical twins. Similarly here, two are different colors, they are free Martin. They were born like this with the single placenta with bifurcated, I mean the two umbilical cords joining the same placenta. So it was found that in the free Martin calf, if the skin graft is done between these calves, even though they are developing independently like this, they will accept the graft. Okay. And in the case of human beings also, the fratellins, if they have a single placenta and connected to that single placenta with umbilical cord, those fraternal twins also can exchange the grafts very easily because they will have developed a tolerance to each one's antigen, because during the process of development itself, they were exposed to the antigens of both of them. Okay, both of them, so that they will develop tolerance to each other. That is what I'm saying here. Color is different, that shows that they are dizygotic. Okay, and they can exchange the graft. Why? Because they are free Martin, because they were developing like this. So from this it is understood that in the early stage of development, if the fetus samples the antigens of the parents, and both the parents antigen, if the fetus comes in contact, fetus will develop tolerance to both the parents antigens and can accept the grafts from both the parents easily. That is what proved. So therefore, one of the experiments that was done and that won a Nobel Prize, not in the human beings, the experiment was done in mice. I will explain about that experiment little later. Before that, there was one more experiment done, which is a wonderful experiment, and that is called as development or production of allophilic mice. Allophilic mice by a person called as Beatrice. Okay, Beatrice Mintz. The experiment is called as Beatrice Mintz. And this person developed this allophilic mice, which is also called as tetraparental mice. Tetraparental mice. This is different from normal genetic cross. In the normal genetic cross, if you were to cross, you would have taken, now here there are two different strains of mice. One, that is C57BL6 mice, that is black in color. Another DBA2 mice, that is white or albino in color. And you know very well, albino is recessive to black. Now what you would have done in genetic cross, you would have taken this mice, would have crossed with this mice. All right. So it is biparental. That is, one mice would have been male, another female, and you would have got a hybrid. Why? Because both these are of two different genetic constitution. One has black pigment, another white. You would have got AB hybrid. In the AB hybrid, you would have got black color expression in the F1 generation. This is simple fundamental of Mendelian law, first law. Okay, so where only male and female that were crossed, and F1 generation will be AB hybrid. Now this allophilic mice is different. Let us consider the C57BL mice, that is black mice, as A strain, and DBA2 mice, that is albino mice, as B strain, for our simple understanding purpose. Once we mention these, the genetic constitution of the mice, it is more than sufficient. Then you can use the code as A and B strain. That becomes easier for understanding. Now what they have done here, Beatrice Mint, what she did is she crossed these two inbreeding mice, means they were genetically close to each other, male and female. She crossed and obtained the blastocyst. Okay, blastocyst. They have shown by color, just for your understanding purpose. Please remember that the black will not be there in the blastocyst. It is only for your understanding. Okay, and in the examination, there is no need for you to draw the mice and all these things. You just mention first the strain of the mice, that is C57BL6 mice and DBA2 mice. Then afterwards you call them as A and B strain and proceed with the process like this in the schematic diagram as shown in the schematic diagram. Okay, okay. Now here, what they did, they mated these C57BL mice separately, and obtained the blastocyst, and mated the DBA mice separately, obtained the blastocyst. Now, these two blastocysts, they put it together in the culture plate, tissue culture plate, okay, and treated that with pronase enzyme, okay, pronase enzyme for the separation of these blastocysts. Okay, the blastocysts which are aggregated, they will separate out into individual cells. And after separation, they were put back in another culture. And when they put back in another culture, all these blastocysts came together randomly, they fused. Another way of separation is if you don't use the enzyme, if you squeeze the blastocyst through muslin cloth, using a muslin cloth, which is like a synthetic muslin cloth, synthetic fiber cloth, or silk like cloth, the blastocyst will separate out. That is how one can separate the tissue cells in the animal cell culture. Even if you take spleen and squeeze the spleen through the muslin cloth, all the lymphocytes from the spleen will be squeezed out into the culture medium. For some time, they will be individual. So here they squeeze these blastocysts out into a culture dish, and both the blastocysts were made to come together. Now, what happened here? They aggregate in large masses, and almost they constitute the same number of cells which was there in the earlier parental combination. That is, A had the blastocyst, B also had the blastocyst. If you count the number of cells there, same number of cells will be there in the fused chimeric blastocyst. This blastocyst is called as chimeric. Now, this blastocyst is different from AB hybrid. The reason being, here equal number of A and equal number of B strain cells will be there. Equal number of A and equal number of B strain cells will be there. Therefore, the A strain 2N number will be there. B strain 2N number will be there. So therefore, this is called as tetraparental allophilic mice. Otherwise, in the AB hybrid of the genetic cross, in the Mendelian cross, you would have had n number of chromosomes of A, n number of chromosomes of B, constituting 2n number. Okay, now here also it is 2n plus 2n. Okay, 2n number of chromosomes of A, 2n number of chromosomes of B. They are there separately in B cells and A cells. Okay, and they have formed a chimeric blastocyst. This chimeric blastocyst, when you transplant into a foster mother which has been made to receive that blastocyst, that foster mother will give birth to the pups, and they will have the expression of both the pigments because of the mosaicism. Here it is a mosaic of A and B strain mouse. Remember that. It is a mosaic. There is no question of dominant and recessive expression. If it was a hybrid, then the A being the black color would have suppressed the expression of B, and it would have been dominant, and the entire mouse in the F1 generation would have been black. Therefore, this allophilic mice will express both the pigments, and it will have equal expression of both the genes, which you can call that as a codominant expression, where you have forcefully brought the two different blastomeres together of two different strains of mice, and both the blastomeres developed into a mouse, which will have now 2n number of constitution of A, 2n number of constitution of B. That allophilic mice, tetraparental mice. Okay, here are some of the examples. Black and white crossed. See the pigmentation in these mice. This is the allophilic mice. Okay. Now, what will happen now when a allophilic mice is produced? Okay. So, a allophilic mice. Now you can do the transplantation experiment. Okay. You have A plus B. We will not be representing that as AB. Why? Because genetic constitution of A as well as B is expressed. This allophilic mice, chimeric mouse, is represented as A plus B mouse. Suppose you transplant the skin from the allophilic mice, okay, into the parent, okay, from this allophilic mice, you transplant into a parent, that is A or B. The parent will partially accept the graft. Why it will partially accept the graft? Because it has the genetic constitution of one of them in equal number. So when you transplant the skin of allophilic mice A plus B mouse, parent, the parent will partially accept the graft and partially reject it. And due to partial rejection, the graft on the skin will look like worm-eaten pattern, as though it is eaten by a worm. It is just like worm-eaten pattern. It is the word used for the partial graft rejection of the skin. Then you can do another experiment. That is, when the skin of A or B, that is parent mouse, okay, skin of A or B, parent mouse, is transplanted into A plus B. A plus B is the chimeric mouse, that is allophilic mouse. A or B, when you transplant it into the allophilic mice, the graft is accepted. Why? Because the genotype of A, genotype of B, both are equally there in the allophilic mice. That is why that allophilic mice will not reject the graft of both the parents, because it is the product of fusion of the blastocysts of both the parents. And the tetraparental blastocyst will have the genetic constitution of these two parents, plus will have the genetic constitution of these two parents. And that allophilic mice will accept the graft of both the parents. So that is what called as tolerance to the different genotype. How it was induced? Because in the embryonic condition itself, you produced a different kind of a hybrid by fusing the blastocyst of A and B strain mouse, and you got A plus B, that is chimeric mouse. That genotypic constitution of that mouse is capable of tolerating, okay, the skin graft, okay, of the both the parents. Now, on the contrary, if you had produced a mice by crossing these two mice, black with albino, you would have got AB. And that AB mouse, okay, if you had obtained, and when the AB mouse graft, that is the hybrid mouse graft, skin graft, is transplanted to A or transplanted to B strain mouse, that is parent, AB hybrid, genetic hybrid this is called as, because n number of chromosomes of A, n of B, together it is 2n. If it is transplanted into A and B, it is completely rejected. Different from the first example here. In the first example, A plus B mouse transplant into A and B, partially rejected, partially accepted, resulting in worm-eaten pattern. But in this case here, the AB mouse, when you transplant the graft to A or B, it will be completely rejected because there is no tolerance. The genetic constitution of this AB hybrid is different from the parent A and parent B. Similarly, vice versa. If you transplant the skin of A to AB, it is rejected completely. Similarly, B to AB, it is rejected completely. Whereas in this case here, the skin of parent, that is parent both A and B, is accepted by the allophilic mice, accepted by the allophilic mice. So that is what a simple experiment by Beatrice to explain the immune tolerance. Now, how the tolerance developed in these mouse is because they were exposed to the antigens of both the strains of mouse during the developmental stage. Even though both the strains are genetically different, because of the exposure of antigens at the embryonic condition, at the early stage of development, both the mouse, that is having different genetic constitution, developed tolerance in the allophilic mice for the graft acceptance of both the strains. Okay, then the Nobel Prize winning work was done by Bellingham and Peter Medawer. Bellingham and Peter Medawer experiment, we will go back to the same, that is A and B mice only. But here it is different. The experiment is different. What they did is A and B mice, okay, they inbred these mice and they in fact transferred the lymphoid cells of A mice, okay, that is infusion. Transfer means infusion. So A mice, that is black, let us say black. A is black. They, after inbreeding, they transferred the antigen of A mice into the neonates. Neonates means newly born, the pups of the inbred B mice, that is B strain. These were crossed, inbred mice crossed, the pups were born to it. To the pups of this B strain, the antigens of the A strain from the pups, that is some amount of lymphoid cells, means you take the blood from the A strain and infuse it into B strain as the B strain neonates are born. And that is what the experiment did. That is, neonates received the lymphoid cells of A strain. So now B strain has received at an early stage, okay, of its growth, the lymph cells of the A mice. Then these mice were allowed to grow. After they are grown, the skin graft from the A was transplanted onto B. Please try to keep a track of this experiment. The lymphoid cells of A strain was transfused into the B strain at the neonatal stage, means young newly born mice at that stage. The immune system is not yet fully developed in the B strain mice. It is yet to develop. At that time, it has been exposed to the antigens of A strain. Now the B strain mouse grew up. After it has grown, to which the transplant of A was done, skin transplant. What would have happened in the normal circumstance? In the normal circumstance, B strain would not have accepted the graft of A because both are genetically different. MHC complex in both these are different. The skin graft would have been rejected, whole rejection or complete rejection would have taken place when the skin of A was transplanted to B. It would have been rejected. But in this experiment done by Bellingham, the B strain will accept the graft of A. It will not reject the skin. Why? It was exposed to the antigens of A strain before its bone marrow matured and completely developed. So its immune system was exposed to the antigens of A at a very early stage. Okay, and then we had seen, after the first graft, first rejection reaction, then when you do the second graft, the second rejection reaction. The second rejection reaction is generally very fast. We had seen first rejection reaction would take almost about 14 days. But second rejection reaction will take within 6 days. So they wanted to see whether the B which accepted the graft, first graft, would reject the second graft when you do it by developing immunity against that. They wanted to see. They performed many skin grafts of A onto B, and it was found that B mouse, as many grafts were done of the A, accepted it. It didn't reject it. There was no second set of rejection reaction. There was no immunity against A. Okay, B mouse had developed tolerance to the antigens of A strain, and graft was accepted. The reason being, A strain mouse antigens were exposed to B strain in the neonatal stage. They were transfused. Therefore, the B strain had developed tolerance to all the antigens of A strain. But now you do a reverse graft. Because A strain was not exposed to the B. A was the donor. What you do is take the skin of the B mouse and graft it onto A strain. Since A is not exposed to the antigens of B strain, the A will not accept the graft. Okay, A will not accept the graft. It will completely reject it. In the first set of rejection reaction, the rejection takes place within 14 days. And if you do the second graft of the same B strain onto A, the A will reject the graft immediately, which is non-tolerance. Means A was not exposed to the antigens of B neonatal condition. Therefore, it is not developing tolerance. It has not developed tolerance. Therefore, it will reject those antigens. Whereas in the case of B, since it was exposed to A strain antigen, it has accepted it. Okay, so then they went one step ahead and told, but the experiment was not allowed to be performed in the human beings. They did the experiment even in the mice at the stage when the pups were developing in the uterus. What they did is they took the antigen of A and injected that into the umbilical cord of the developing B strain pups in the uterus of the mother and sutured back the uterus. The lymph cells or lymphoid cells of the strain were injected into the umbilical cord of the B strain mouse that was developing in the uterus of the B inbred mouse. And they allowed those fetuses to develop into pups. And the pups were born. And after the pups were born, they received the transplant of the skin of A, and the graft was accepted. And this experiment was slightly different from the previous experiment. In the previous experiment, B mouse was allowed to give birth to the pups. As soon as the pups were born, they were injected with the lymphoid cells of A strain. But in the second experiment, what they did? Before the pups were born, in the uterus itself, through surgery, opening the uterus and injecting the antigen of A into the the cord of the B strain pups, and then suturing it back, allowing the pup to develop completely the entire gestation period and then be born. After they were born, then they were allowed to grow. Then A strain mouse skin was grafted onto them. It was accepted. So then they came out with a hypothesis. If this could be done in human beings, human fetus, if it is given as the development is taking place in the mother's uterus, the antigens of both the parents, even though the fetus is the n number of constitution of parent A and parent B, The fetus, after it's grown, after it is born, it will not accept the graft of both the parents easily because the constitution of the fetus will be different. It is like a hybrid. They said that if through the umbilical cord using sonography, if the blood of A parent and blood of B parent is fused into the umbilical cord after the birth of the child, when the child grows, later if it needs the organ for some reason, it can accept the organs of both the parents easily without rejection. Without rejecting, it can accept the organs of both the parents. And I had already told you in the transplantation that there is no possibility of getting 100% match for the siblings that are born to the same parents, nor to the child which is born to the same parent. There is no possibility of getting 100% match. Therefore, the grafts, when done, they are going to be rejected. And therefore, need to take precaution. And you can get 85% match, but remaining 15% you have to win by administering immunosuppressive drugs. But through the Beatrice-Mintz experiment, as well as through Bellingham's and Medawur's experiment, we have come to know now that if the antigens are exposed to the developing fetus at the early embryonic stage, that fetus will accept that antigen by tolerance. But remember, such type of tolerance should not take place in the fetus for bacterial and viral antigens. Therefore, a pregnant woman has to take utmost care to not to expose the fetus to any bacterial antigens if they can diffuse through the placenta. If those antigens go through the placenta to the fetus will develop tolerance to those antigens. Then when that child is born, when the child is exposed to that pathogenic organism in real life, the child will not be able to fight the disease caused by that pathogen because of the tolerance developed against it. And it will not recognize that antigen as foreign antigen, but the disease causing symptoms, one cannot develop tolerance. Only to the antigens one can develop tolerance. And imagine a pathogenic antigen was exposed to the developing fetus in the mother's womb, and that fetus, after it is born, gets exposed to that pathogenic bacteria, the fetus will not survive, or the child will not survive. Therefore, one need to take care during the pregnancy to prevent the development of tolerance against pathogenic organisms. And mother should take care that she is not exposed to the pathogenic organisms. Okay, and here I've explained to you about this tolerance mechanism, and you have seen in the newspaper, and there were women who were pregnant exposed to coronavirus. And being exposed to coronavirus, these women might have transferred the soluble antigens of the coronavirus through the placenta to the fetus. And that fetus will have tolerance to all the soluble antigens of the coronavirus. And when that fetus is born, later in its life, after it grows, in future, if there is coronavirus and that child which has developed has not been immunized or vaccinated for coronavirus, there is every chance that the child may succumb to coronavirus after it is grown as an adult. It is also shown that majority of young children are not susceptible to corona infection because they do not have sufficient number of ACE receptors. receptors are not well developed in the case of young children, and only in the adult ACE receptors are well developed. Therefore, adults are succumbing to the coronavirus infection. Young children are not succumbing. But few cases are there where young children also became infected, and they had comorbidities, and that is the reason they were infected, whereas normal young children are not infected. But doesn't mean that in the later part of their life, when they become adult, they will not develop coronavirus infection, because coronavirus is highly mutable. The vaccine that is developed against coronavirus may work now, but later, after few years from now, it may not work. Why? Because the virus is a highly mutable type, just like the HIV virus. So finally, if I have to conclude, that tolerance is a phenomenon where immune cells develop tolerance to the antigens if they are exposed to these antigens at the early stage of their development, in the young stage, that is in the neonatal condition or in the embryonic condition. But tolerance should not develop against pathogenic organisms or pathogenic antigens. It is dangerous for the life of an individual, because the pathogenic organism, when it infects, it will cause disease symptoms, and disease may be highly detrimental, and pathologic condition of the disease may be highly detrimental. So for today, I'll stop here. Thank you very much.